So I would like to welcome you all, and I would like you to join us on this journey through the microbial dark matter of the moose gut, or actually the moose rumen. Okay, to reach a sustainable society, we need to reduce our dependence on oil and coal. And in order to do so, we have to use our renewable resources more efficiently. And one such resource is plant biomass. And in Sweden we have a lot of biomass, especially in the forest. But plant biomass could also come from agriculture, and actually also from waste. We have a lot of food waste that we can use. Now, through various processes, those could be biotech, chemical or thermal processes, we can convert this biomass into bio-based products. We do that today. For instance, we can make paper and food, but we can expand this range of bio-based products to include also green chemicals, biofuels, packaging, material, pharmaceuticals, and textiles. So when I say we need to use this more efficiently, I mean we should use 100% of the biomass. I can take an example. In the old pulp mills, we used the cellulose, about 50% of the biomass, to make paper. And we didn't care about the rest, about the lignin and hemicellulose. But in the future, we want to make bio-based products out of that too. So we use 100%. We want to utilize the biomass to 100%. Now, Anders and me, we are doing research on biotech, trying to find biotech solutions, converting this biomass to bio-based products. So what is plant biomass? Well, actually, the main portion is carbohydrates, 75%. And here I've just listed some of the most common plant carbohydrates. Again, cellulose, that we make paper out of. We have hemicelluloses like xylon and mannan. We have pectins, and we have starch. Now, the plants make a lot of this biomass through the process of photosynthesis. So on land, about 120 billion tons per year. And in the oceans, the algae contribute with another 50 billion tons. So we have a lot of this renewable resource, okay. And in nature, this biomass is built and deconstructed by enzymes. So the question is, can we use these enzymes to convert the biomass into these bio-based products? Now, the enzymes are also interesting from a sustainability aspect. So what are enzymes? Well, enzymes are very specific biological catalysts. They are proteins that accelerate chemical reactions. And because they are specific, we can reduce chemical consumption. And we can reduce unwanted side reactions, toxic compounds, for instance. And enzymes can carry out reactions under mild conditions. We can lower the temperature of the process and thereby saving energy. And also, as I said, enzymes are proteins, so they are biologically degradable. Now, the main portion of biomass, you remember, is carbohydrates. So the enzymes we are interested in that degrade or modify or build the carbohydrates, they are called carbohydrate active enzymes. Here you can see just an enzyme, the 3D structure of an enzyme. And here you can actually see an enzyme in action. This is a cellulase degrading a cellulose chain, releasing cellobiose, a molecule of 2 glucose sugars. So, the dominating biofuel today is bioethanol of the first generation. So that is made of crops like corn, but also sugarcane. And here we use enzymes to degrade these carbohydrates, starch and sucrose. Now the thing is, these carbohydrates are very easy to digest using enzymes. So we have in our saliva amylases, starch-degrading enzymes, and we have used enzymes then for thousands of years to produce wine and beer. So it's very easy to switch from drinking the ethanol to put the ethanol in the car. But first-generation biofuels has been criticized because should we really use food for biofuel production? Now the alternative would then be to use plant parts that we don't eat, or plants that we don't eat. That could be trees, wood, that could be waste from agriculture, wheat straw, it could be energy grasses. Now these types of plants and tissues are consisting of something called a material called lignocellulose. Lignocellulose has three main components. Again, cellulose, a carbohydrate, a long chain of glucose units. We have hemicellulose, also long chains of different sugars with side chains. And we have a third polymer called lignin. What is special with this lignocellulose is it's a material designed to resist degradation. You can think of an oak living for thousands of years. Okay, so the challenge here is we must be able to degrade this as efficiently as this one. And we're not there yet. We can degrade this, but not cost effectively. So we need to, in order to compete with fossil fuels, we need to find more efficient enzymes for this process. So this is the big challenge. Easy to digest, difficult to digest. And we want to solve this problem. All right. So where can we find these new efficient enzymes? Well, we should look in biomass degrading environments. For instance, we can look in the compost, but we can also look in the guts of animals that eat plants, herbivores. Or we can look in soils. Now, the traditional way to look at fungi and bacteria in these environments is we try to isolate it and cultivate it in the lab. And that's fine. You can discover new enzymes and so on. But the problem here is we are limited only to the microbes that we can cultivate in the lab. And that's actually just a small, small fraction of all the microbes out there in the world. It's been estimated that more than 90% of the microbes cannot be cultivated in the lab. And those microbes that we cannot cultivate in the lab has been termed then microbial dark matter. But now, with new technology, with a method called metagenomics, we can actually also look at these microbes that we cannot cultivate in the lab. And now comes Anders.
Yes. So with metagenomics, metagenomics is the direct analysis of DNA from environmental samples. And with metagenomics, we can analyze any kind of sample, such as soil, compost, seawater, or the content of an animal gut. And the basis is that you lyse all the microbial cells in your sample, you extract DNA from this pool of cells, and then you sequence millions of short DNA sequences from this pool of DNA using a high-throughput sequencing instrument that are now available. And having this catalog of short sequence reads, you can compare those to genomes in databases, and by that you can get a list of what type of organisms are present and approximately what type of genes they carry. But the nice thing is that you can also investigate totally new life that haven't been cultured before, because you can puzzle these short DNA sequences together into longer genome fragments, and then using advanced algorithms that we have and others have developed, we can actually figure out which of these genome fragments come from the same species. So we can reconstruct more or less complete microbial genomes. And these genomes are then a great resource for finding new enzymes for biotech. Let's see. In Sweden, we have about 300,000 moose walking around in our forests. And these moose, especially during winter, eat a fiber-rich diet of twigs, branches, bark, etc., of a variety of trees. And this is, of course, kind of a big problem for the forestry industry in Sweden. They do cause significant damage, especially to young trees. But we see a window of opportunity here, because just like, for instance, cows, the moose is a ruminant, so it uses its first stomach compartment, the rumen, to digest complex polymers and carbohydrates from the plants. And the rumen is a virtual anaerobic bioreactor brimming of microbial life. So there are billions of microbes there that digest the plant fibers into food particles that the moose can utilize. And so far, very little is known about the microbes in the moose rumen. So we thought that with metagenomics, we can get a new picture of this ecosystem and maybe find interesting enzymes. So we analyzed the content of 6 moose rumens from moose that were shot during the annual moose hunt a few years ago. And these 3 of the moose came from Småland and 3 from Öland. And we sequenced some 100 billion base pairs of DNA from these samples. And just comparing the short DNA reads that we got with reference databases, we could kind of get the profile of what microbial taxa we had in those samples. And like most other vertebrates, the samples were dominated by Bacteroidetes and Firmicutes, 2 major bacterial phyla. So from that perspective, there at the kind of over high level, the microbial content is quite similar to ours and in the cows, etc. What we also found, however, was quite high abundance of spirochetes, and spirochetes are very uncommon in vertebrates, but they're instead common in termites that are wood-eating insects. Okay, but having all these short reads, we were also able to puzzle them together into microbial genomes. So we got something like 99 more or less complete microbial genomes. And here to the left, you see a phylogenetic tree of those genomes. And the parts of the trees are colored according to the bacterial phyla that they represent. And again, it's dominated by the Bacteroidetes and the Firmicutes. And to the right, you have instead the profiles of the carbohydrate active enzymes in those genomes. And by investigating these profiles, we could deduce that some of these microbes are very specialized in certain types of substrates, while others are more generalists. And while a few of the microbes, I should say, are quite close relatives of microbial strains that have been isolated from the cow rumen, most of them represent species that have never been analyzed or cultured before. So these are novel species and genera. And as such, they of course they have the potential to contribute to enzyme discovery. And we have then analyzed in detail the enzyme portfolio in these genomes. Now Henrik will tell a little bit more about that.
Yes. So we have these 99 genomes. So we have screened them then for genes encoding these carbohydrate active enzymes. And we found a lot. So thousands of interesting enzymes. For instance, we have more than 6,000 of these glycoside hydrolases. This class of enzymes includes cellulases that degrade cellulose. We have xylanases that degrade xylan. We have mannanases. Basically, we have all enzyme activities we need to degrade all types of plant carbohydrates. Also, in these other enzyme classes, we have interesting enzymes. So in here, we have targets with interesting putative properties that we would like to continue to study. These could be the new efficient enzymes. But it's actually not just the enzymes themselves that are interesting, also how they are organized in these microbes. So in these Bacteroidetes that Anders talked about, they organize these K-enzyme genes in gene clusters called polysaccharide utilization loci, or pools. So here, all enzymes are sitting together, and they are working together to degrade a specific carbohydrate. So here we have one package, a toolkit that we can use, for instance, in biorefinery processes. So here we have an example of such a pool predicted for xylane degradation. But we also see in these gene clusters unknown genes. And these are, of course, interesting because they could represent totally new enzymes. So these are also interesting targets for us for further investigation. In other types of bacteria, the K-enzymes are organized in cellulosomes, in Firmicutes or the Ruminococcus. Now, the cellulosome is like a protein scaffold on the outside of the bacterial cell. So here the enzymes are docking to other protein modules, so they can orient themselves on the outside of the cell in a way so they can efficiently degrade polysaccharides. So these are also interesting to analyze. Now, maybe the biggest discovery we made in the mouse rumen was that we actually saw that in some Bacteroidetes genomes what looks like a mix of this cellulosome and the polysaccharide utilization loci. So we don't really know the function of these yet, but it may represent a novel strategy for plant carbohydrate degradation. So this is definitely something we would like to continue to work with. So how would we continue this project? Well, in the end, we would like to develop new enzyme cocktails for degradation of plant biomass. So we have these interesting mouse rumen enzyme genes. We cannot look at all the thousands of them, but we can look at 10 up to 100 maybe. And what we need to do is to get the protein. So in a host like E. coli, we let them produce the protein so we can look at the function of the enzyme, determine the catalytic properties. And if that is promising, then we can include the new enzyme in these enzyme cocktails. But perhaps we need to improve a certain property so it fits into the process. But after that, we can include it in the enzyme cocktail. So in the final step, in the enzyme cocktail design, we can test different enzyme components. We can test different enzyme ratios and try to optimize for different feedstocks. So we have enzyme cocktail that will degrade spruce or pine wood chips, another that degrades wheat straw, for instance. And these feedstocks may have also been pre-treated in various ways. We could also try to design these enzyme cocktails for complete degradation down to the monosaccharides, or a selective degradation, where we actually try to fraction out some of the oligosaccharides. But in order to do this, we need funding. Otherwise, this project will end here. That would be kind of sad. So, I would like to thank you for listening and also acknowledge all the people involved in this mouse rumen project. So we have our colleagues at KTH, Olof, Johannes and Nino. We have people in Marseille, France. We have other people in Uppsala. And we also have a group in Luxembourg. And also we would like to thank Formas for funding. And thank you.